Okay, please go ahead.
Okay. Welcome to today's Immune Zoom. It's my great honor to introduce today's speaker, Dr. Soman Abraham, who is currently the Grace Kirby Distinguished Professor of Pathology in Duke School of Medicine and also in Duke National University of Singapore. So first, just some brief background about Soman. So he received initial training in microbiology. Soman obtained his bachelor and master degree in Ahmadu Bello University in Nigeria, which was followed by a PhD in the University of Newcastle, UK. Then Soman came to the United States to do a postdoc with Edwin Beachy in the University of Tennessee. He also started his own lab in Tennessee in 1986 and moved to Washington University in St. Louis and finally to Duke in 1997. Soman's lab has made numerous important discoveries on pathogen-host interactions. He has also been interested in mast cell biology, which I think is a focus for today's talk, actually for over 3 decades. So mast cell is, I think, a long understudied immune cell type, which is traditionally associated with inflammation and asthma. I think one of Soman's most exciting discovery is that mast cells also play a key role in immune surveillance during bacteria and viral infections. And this is mediated by modulating both innate and adaptive immune responses. In view of these powerful immune regulatory function of mast cells, Soman's group also discovered small molecule activators of mast cells that can remarkably enhance antibody response to the vaccine antigen in the host. And his studies revealed for the first time that mast cell targeting molecules were potent vaccine adjuvants. So beyond these excellence in science, Soman is also dedicated to mentoring and nurturing young scientists. He has mentored over 50 PhD, MD students or postdocs. He served as a graduate student advisor at Duke Pathology for over 20 years. And personally, I also owe many gratitude to Soman. So when our lab initially started working on mast cells, I know basically nothing about it, and it is very difficult to find a mast cell expert. And luckily, I approached Soman, even we had not met before, and but Soman is extremely kind and provided many helpful suggestions, which is one of the key reasons that we can continue our research. So thank you, Soman. Without further ado, the floor is yours.
Right. Thank you, Shirley, and also want to thank Xuan Wu for inviting me to share some of my work with all of you. It's an honor, and I will proceed straight to the topic. As Shirley said, mast cells are understudied, and so we took up this series of studies a long time ago. And as I was reviewing the slides, I realized how fast time has gone. I'm hoping I will be able to successfully educate you on some aspects of mast cells that you did not know before. All right, so mast cells, as Shaoli had indicated, are one of the most enigmatic cells in the body. It was first discovered in 1877 by Paul Ehrlich, and he called them Mast Zellen, which in German means well-fed cells. And that was because of the large number of granules that these cells seem to encase. And it was not really known what they did. But for a long time, mast cells have been mostly associated with disease, allergic diseases, including asthma, rheumatoid arthritis, bullous, pemphigoid, multiple sclerosis, and so on. So they were essentially considered as really detrimental cells. But what is the physiological role of mast cells? If mast cells were exclusively doing bad things, they would have been removed through evolution. The fact that they still remain in our bodies in spite of its negative role would suggest that whatever they do is important. And there are several properties of mast cells that suggest a role in immune surveillance. One is their location. This is a picture of the skin, and here are mast cells. So they're sitting right underneath the epithelial layer. So they are one of the first kind of immune cells to see incoming pathogens as they break through. And this is a picture of the bronchial epithelium. And you can see a lot of mast cells sitting underneath the epithelium. And in this particular case, you can see a particle coming through, penetrating the epithelium, and the mast cells interacting with it. And mast cells have certain unique properties that enable them to play a key role in what we hypothesize is a role in immune surveillance. That is their capacity to degranulate. So when they get activated, within seconds, they degranulate, releasing all their granules, which are rich in inflammatory mediators. So this happens within a few seconds to minutes. And then after that degranulation, then the mast cells undergo de novo synthesis and then continue to secrete various cytokines and so on. So there's an early response, which involves release of the granules, which contain histamine, proteases, and various pre-stored inflammatory mediators, as well as then a de novo synthesis phase, which is initiated from about 3 hours and can go on for about 12 hours. So there is a sustained release capacity of mast cells that can prolong inflammation. And in addition to that, mast cells have the unique capacity to regranulate after degranulation. So after activation, they can release the granules, and then in the same situation, they don't die off, they remain alive and then regranulate at the same site. And so they can potentially undergo multiple rounds of degranulation. The other unique property of mast cells is the fact they're sitting next to blood vessels and the lymphatics. So this is an image of a whole mount of the ear of a mouse. The green are the lymphatics, the red are the blood vessels, and in between you can see in blue are mast cells. And if they don't show very well on the screen, if you go to the right hand side, this is a cross section of the same ear, and you can see the blue are the mast cells, the green are the lymphatics, the red are the vasculature. And you can see mast cells are next to the vasculature, and so potentially can regulate what is happening in the vasculature. And in addition to that, mast cells have the capacity to directly recognize pathogens. So on the surface, they have TLR4, the various TLRs. They also have the capacity of recognizing microbes that have been coated with antibodies and complement, because they have receptors on the surfaces that can recognize complement-coated bacteria, or in this case here, IgE associated parasites. So they can also recognize that. And in addition to direct interactions, as I said earlier, this is a picture of a mast cell associating with a bacteria. You can see them producing protrusions that seem to be binding the bacteria. Now, another point I want to make is infectious diseases are one of the leading causes of death, even in the United States, which is a well-developed country. So this is data from 2020. When you look at the major causes of death, we have heart disease and cancer, but after that you have COVID, and then if you include chronic lower respiratory infections and influenza and pneumonia, you actually get infectious diseases as being the major cause of death in the US. And you can imagine in a developing country, that is even more pronounced. So clearly, infectious diseases are a major cause of death for mankind. So based on all of that, we hypothesized that mast cells, because of their location, the capacity to release mediators, the location near the vasculature, may play a very important role in the early innate immune response to pathogens. So if you have the epithelial barrier here, you have the pathogens breaking through. Mast cells, which sit out here, have the potential to recognize the pathogens, release mediators that can bring out neutrophils and other immune cells to come and combat the infection. In addition, they produce proteases which can break the tight junction around the blood vessels, allowing leakage of fluid, which also will include antibodies, complement, and so on, which can coat the bacteria. So in addition to getting the recruitment of critical cells, they also can get antibodies and complement to leak out to then attach to the infecting bacteria. So, so based on all this, we did our initial study, which was done many, many years ago, where we showed for the first time that mast cells had the capacity to recognize bacteria and bring in neutrophils. And it was actually mast cell TNF that was critical in bringing in the neutrophils. And at that time, Professor Stephen Galley, a leader in the field of mast cells, indicated, or in his review, he indicated that the mast cells had two phases. There was a detrimental phase where it was involved in asthma, but then a beneficial phase where it was protecting us from infection. So that was what was thought of in 1996. But since then, what I want to tell you is our discoveries since then have tended to suggest that not only do mast cells have two phases, they actually have many phases. And I'm hoping to convince you of that as we proceed. So as indicated earlier, we were able to show that mast cells, which sit out in the epithelial barrier, are able to recognize viruses and bacteria, release mediators, and these would result in the recruitment of neutrophils from the blood to the site of infection. And in the case of viruses, say, for example, dengue virus, when you have a mosquito injecting the virus into the skin, you would have NK cells and NKT cells being brought there by mast cells. So mast cells were very important in the early recruitment of the critical immune cells to clear the pathogen. But what I want to focus today more so is their role in turning on the adaptive immune response. So while they're doing their recruitment of immune cells to clear the pathogen at the site of infection, which is here, they also play a key role in recruiting critical immune cells to the lymph node, which is the draining lymph node, which is also the epicenter of the adaptive immune response. So typically, the adaptive immune response is initiated when dendritic cells, which have sampled the pathogen, are then recruited or trafficked into the draining node. And at the same time, T cells, which are in the circulation, extravasate and then are sequestered in the lymph node. And the interaction between the dendritic cell and the T cell is critical to turn on the adaptive immune response. So what I want to tell you today is mast cells are also critical in that aspect. And a lot of that data comes from studies we undertook where we injected bacteria into the foot pads of mice and studied the popliteal lymph node, which is the draining lymph node. And we were able to show that mast cells were critical in the coordinate trafficking, that is the simultaneous trafficking of dendritic cells into the lymph node and also T cells from the circulation into the lymph node. Them coming together was critically mediated by mast cells. So the studies we undertook at that time involved use of wild-type mice, mast cell deficient mice. These are mice that are deficient mast cells due to mutation in c-KIT and the receptor for stem cell factor. And then we had a third group, which is mast cell deficient mice that we had repleted with mast cells. And in some studies, we had mast cell deficient mice that we repleted with TNF knockout mast cells. So we wanted to show in those specific cases the critical role of TNF. And we compared what was happening between these three groups of mice after E. coli injection into the footpad of mice. And then we were studying dendritic cell and T cell migration into the draining lymph nodes. So our data show that when you look at the recruitment of dendritic cells into the lymph node, we see in the mast cell deficient mice, very little recruitment, but in the mast cell deficient mice that have been repleted with wild type mast cells, we have very good recruitment of dendritic cells. And in the mast cell deficient mice that were repleted with mast cells from a TNF knockout mice, we see very little recruitment, indicating that mast cells and specifically mast cell TNF was critical in the recruitment of dendritic cells into the lymph node. And when you actually look at the lymph node after 24 hours, we see this huge increase in size compared to the controls. And this growth in size is largely attributed to the migration of T cells and B cells into the lymph node. And here is looking at the growth in size of the lymph node following the bacterial infection. So this is what you see in the wild type mice. This is what you see in mast cell deficient mice that were repleted with wild type mast cells, and you see very nice growth in lymph nodes. And then this is what you see in a mast cell deficient mice that were repleted with mast cells from a TNF knockout mice. Okay, so here again, we are making the point that mast cell TNF was critical in the lymph node hypertrophy, the growth of the lymph node following sequestration of T and B cells into the lymph node. So what we're saying that is that mast cells sitting out here was regulating the migration of dendritic cells from the tissue here into the lymph node, and at the same time regulating the sequestration of T cells and B cells into the lymph node following infection. So how was this happening? And all this was happening in 24 hours. So what we did was looked for the chemokine CCL21. So CCL21 has been shown to be the critical chemokine produced in lymph node to bring T cells out of the circulation and also dendritic cells from the site of infection. So we assayed for the presence of CCL21 over time following infection. And we see that by 3 hours, we see significant amount of CCL21, and this seemed to progressively increase with time. And then when we look to see if we use antibodies to CCL21, could you inhibit the recruitment of dendritic cells into the lymph node? And the answer is yes. And when we use the antibodies to CCL21, could you inhibit the sequestration of lymphocytes in the lymph node? Yes, we're able to block that. So CCL21 appears to be the critical chemokine produced in the lymph node that was responsible for recruitment of both dendritic cells and lymphocytes into the node. And this is looking at the increase in CCL21 expression in the lymph node, where we see in wild type, in SASH mice, mast cell deficient mice repleted with mast cells from a wild type mouse, and we see very nice CCL21 expression. This is what you see in a mast cell deficient mouse. This is what you see in a mast cell deficient mouse repleted with mast cells from a TNF knockout mouse. And again, here you don't see much a CCL21 expression, indicating that it was mast cell TNF responsible for the increase in CCL21 produced in the lymph node. And when you actually stain for CCL21, you can see here following E. coli infection, there's a very strong CCL21 localization in the node as against the control. So to further make the point that it is actually mast cell TNF, so mast cell TNF that was responsible for the upregulation of CCL21, we assayed for TNF production in the lymph node. And we see that by 3 hours after bacterial infection in the footpad, the levels of TNF has gone up to significant levels by 3 hours. And what was interesting is when we actually looked in the lymph node for message for TNF, we didn't see message for TNF, although we saw message for TGF-beta. So there was no message indicating that the TNF seen in the lymph node was produced from somewhere else, which, as we suspected, is what we hypothesize actually is that it is the TNF from the site of infection that was reaching the lymph node. So what we are saying is mast cells here, following infection, produced TNF, which was then trafficking through the lymphatics, getting into the lymph node, upregulating CCL21, which was then causing the migration of dendritic cells from the site of infection, and also T cells from the circulation into the lymph node. Okay, so that was our hypothesis. So, however, if that hypothesis was correct, then one would imagine that these mast cells here should produce a large amount of TNF to traffic all the distance into the lymph node. And TNF is highly labile. It tends to get degraded within minutes. So how was this being achieved? Because there are not that many mast cells here, and they don't produce as much TNF to come all the way through the lymph node. So actually, to help you visualize what could potentially is happening at these sites. So here you can imagine there's a person here who gets infected in the foot, and you have the mast cells getting activated. So what we are trying to convince you is that here the mast cells were producing sufficient TNF to traffic all the way to the lymph node, and then in evoking changes in the lymph node. And this is a tremendous distance. So it's hard to believe that there will be sufficient TNF to do this. So how can you explain how mast cell TNF was affecting the draining node? So an idea came to us based on looking at mast cells following activation. What we see is, we see a lot of granules, and that is expected when mast cells get degranulated. But what if these granules are not broken down as everybody had assumed? Maybe they are stable, and maybe they could be the carriers of TNF to the lymph node. So a little bit about mast cell granules. So mast cell granules are stored within membranes in the mast cell, and when they are released, they go out, but there's no membrane around them. So, and the reason they're held together is they're made up of heparin, which is negatively charged, and a lot of proteases, which are positively charged. So they're held together by electrostatic interactions, and then the cargo that are carried are held in between. So these are nice spherical particles made up of positively charged and negatively charged compounds, but inside they're carrying cargo like TNF and so on, which then are held together. And we believe that this is how TNF is stored and kept undiluted and protected from degradation. And at the same time, these granules could potentially traffic to the lymph node. And so this is just an image of a mast cell. And as you can see here, when you degranulate, you see the granules coming off. So the mast cells release these granules, but these granules are not breaking down as everybody assumed. They're highly stable. So this is a picture of a mast cell before degranulation. This is after degranulation. And you see a lot of granules out here. But interestingly, we can isolate these granules. And when you look at these granules, they are extremely stable under the microscope. And in fact, they can remain for over 70 days at room temperature, still intact. So they're extremely stable and quite unlike what everyone had suspected. And this is an electromicrograph of a single mast cell granule. This is what it looks like. So the key question is, when you isolate these granules, do you still see TNF in them? So we ran a gel, and this is just recombinant TNF, and on a Western, and we see these mast cell granules contain TNF, right? So even though they are released, the TNF is still kept in there, tightly held. So then we can imagine how these granules enter the lymphatic vessel. So the lymphatic vessels have lot of space in between. So the epithelial cells that line the lymphatics. So the granules can easily slip in and get into the lymphatics and then go with the flow. So there's no active movement, they just go with the lymphatic flow. And we think that is how the mast cell granules are released here and they get into the lymph node. And this is a picture of the mesentery of a rat. And I don't know if you can make out, there are a lot of blue here. And then these are the lymphatic vessels, which seem to service this area. And if you look closer, you can see a lot of mast cells are next to the lymphatics. So the mast cells are very close to the lymphatics so that if they release granules, they can easily get in here and then flow. And this is actually the mesentery again, and this is the lymphatic vessel. These are activated mast cells. And if you actually track the granules in here, you can see them. They're going inside these vessels and they're trafficking. So you can track that. So these mast cells, when they're activated, the granules do enter the lymphatics and get into the lymph node. And this is a cross section of a lymphatic vessel, and these are mast cell granules out here. And I think one of the reviewers asked us to show mast cell granules in the lymphatics, and that's essentially an impossible task. But we managed to get one example where we think we can make out a granule within the lumen of the lymphatic vessel. All right, so we were able to show that mast cell granules were present in the lumen. But we did a more definitive experiment to show that mast cell granules, when they get to the lymph node, they can cause, and especially the ones containing TNF, they can cause a tremendous increase in the lymph node size because of the TNF that they carried. So this is a cross section of the lymph node, I think a few minutes after introduction of mast cell granules into the foot pad. So in this experiment, we collected mast cell granules from a wild type mouse and then from a TNF knockout mouse. So the mast cell granules from both. So in one case, there would be granules with TNF in it and the other with granules with no TNF. That was the only difference between the two granules. We injected that into the foot pad of the mice, and we can see here there's a huge increase in the wild type case of the lymph node size. So the TNF produced by the granules was causing the hypertrophy of the lymph node, whereas granules alone without TNF we didn't see that. So that was definitive proof that it was mast cell granules and specifically TNF in it that was responsible for the hypertrophy. And so what we now believe is that mast cells have the capacity to turn on the adaptive immune response through release of granules, which then traffic to the lymph node, which is the epicenter of the adaptive immune response, to then boost the immune system, right, to fire up the immune system, the TNF getting there, the chemokines produced, and then you get all the immune cells trafficking in. So the mast cells have that capacity to drive the immune cells. And so during infection, that's what the mast cell is doing. So with that in mind, we asked a very simple question. Can we apply the unique properties of mast cell granules to target lymph nodes to boost immune responses? Okay, so there's a major limitation in the vaccine industry currently, and which is the lack of effective adjuvants. There's always a search for adjuvants, particularly when you are using protein antigens, subunit antigens, as against mRNA and all. Even with mRNA, you do adjuvants, you do need, and in the case of mRNA, is the liposomes that they are encased in. So, but for the subunit antigens, you do need adjuvants, and there's a big dearth of that. And because a lot of the vaccine antigens are not immunogenic themselves, so you need some sort of adjuvant. So we hypothesized that what if we create our own mast cell granules where we put in cytokines such as TNF, which is known to boost the immune response, and then as well as put the antigen in the granules and then inject them. So since these granules have the capacity to traffic to the lymph node, that would be an optimal mechanism to direct the antigen to the lymph node. Because what the adjuvants do in all these vaccines is to activate dendritic cells to come and sample the antigen and take them to the lymph node. So here, what if we put them into these granules, mast cell granules, and traffic them to the lymph node, as they naturally would be doing in an infection? So what we did was we then loaded the vaccine antigen into mast cell granules that we have synthesized, and I will tell you how we synthesize them. But the idea was to inject them into the foot pad of mice and then look at what is happening to the lymph node and also to the immune response. And so we made our own synthetic mast cell granules, which was mimicking what you have in the natural case. So in the natural case, we have heparin and the proteases serving as the backbone, and then you have the cargo in there. So what we did was we made our own, where we mix heparin and chitosan, both of which are biodegradable and have been shown in humans to be safe and very efficacious in delivery of material. So we put the two together, negatively charged, positively charged, and then we loaded them with TNF. And so we generated our particles this way. They look like this. They're not as beautiful as the natural ones, but on a lower power microscopy, we see they're about the same size. These are our synthetic granules, and this is the natural mast cell granules. And then this is a little bit about them. This is the natural granules. They are made up of heparin, proteases. This is the molecular weight. The same thing, heparin here, and chitosan, which is very biodegradable. And so we made ours. We loaded it with TNF here, and then and we also actually loaded some of these with FITC to track them. And this is what you see. When you inject them, within 15 minutes of injecting them in the foot pad, you can see that these particles easily go into the lymph node. And they are in the capsular sinuses here initially, but later on with more time, with more time, the granules then go out and move into the T cell, B cell zones, and so on. So they are able to carry the antigen into the lymph node and also carry the TNF, which can activate the lymph node and boost the responses. And this is actually what we show. So if you inject just the particles in here, the lymph node do not grow in size. But if you load them with 0.3 nanograms of TNF, you see more than double the growth of the lymph node. And if you use soluble TNF, such as 1 nanogram or 10 nanogram, both of which are more than here, you barely get much. So if the TNF is put in a particle, the biodegradable particle, it can boost the immune system. Whereas if it's soluble, it's not doing much because a lot of it is degraded and diluted. So this is the way to traffic TNF to the lymph node so that it is very potent. And we actually, when we look at the germinal zones of the lymph node, you see that with particular TNF, we see a lot of growth. This is the B cell zones and germinal centers. And there's a tremendous amount of growth there because of the TNF, which are loaded in the particulate particles. And then we also get very good antibody responses against the antigen. This is IgG, IgG1, IgG2A. Compared to the controls, and also with regard to the avidity of the antibody, which is very important for protection of functional aspects, we do have a much higher avidity seen with the antibodies generated following injection of particles with TNF in it. And we also get protection here compared to the controls. So this is protection. This is against the lethal flu injection where we use hemagglutinin as the antigen. And we also did an experiment where we used the Fluzone, which is a vaccine used in humans, and we gave it Fluzone alone into the mice. And then this is saline. We get a good response against Fluzone, but we get 100 times more efficacious response if we use particulate TNF compared to the soluble TNF. So by loading the antigen into particles along with TNF, we are able to get a much, much stronger immune response. So in addition to boosting the immune response, we can actually alter the nature of the immune response based on what cytokine you put in. So we have been using TNF to boost the immune response, but if you put in a cytokine such as IL-12, which is a Th1 skewing cytokine, you can actually alter the immune response in an animal. So we have a model here where we have a peanut allergy induced in a mouse with a lot of IgE against the peanuts, and then there's a very potent reaction to the peanut allergen when we in this model, the mice have very potent reactions to peanut allergen. But if we vaccinate these mice with peanut allergen along with IL-12, which is a Th1 skewing cytokine, we could actually alter the reaction of these mice in such a way that the immune response was then redirected to a Th1 type of response. So we could re-educate the immune response in a mouse which is already trained to be giving a Th2 and a harmful reaction to giving a Th1 and harmless reaction by injecting the vaccine antigen along with this Th1 skewing adjuvant. So by directing this mast cell inspired granules to the lymph node, we can dictate the nature of the immune response. So I will now switch to another area, and that is, I want to talk to you about mast cells found in a very different part of the body, part of the body most of you don't really think about, and that is the bladder. All right, so the bladder is a very unique organ, and it's lined by epithelial cells, which are very tightly held together. This is the image of the bladder epithelial cells, and they're very tight junctions. And the idea is to prevent urine. When urine comes in, the epithelium has to protect against urine leaking in. Urine contains ammonia, salts, all of which can be toxic to the underlying tissue. So I want to tell you a little bit about what mast cells in the bladder do in this situation. So particularly during infection. So this is so typically infections start with the bacteria getting into the urethra, getting into the urine, multiplying the urine, and then you infect the bladder and you get cystitis. Sometimes it goes all the way up and you get pyelonephritis, kidney infections. Okay, so this is just to orient you. This is a microscopy from the top. So this is the superficial epithelium in green, the mast cells are in red. Okay, so that's what you want to look at. Now, from the side view, this is the superficial epithelium, and then these are the mast cells. Okay, so they're sitting underneath. And so the mast cells are oriented right underneath the epithelium. So what happens, and this is very typical in the urinary tract infections, is when bacteria get into urine, they grow like crazy in the urine, and then they bind to the epithelium. And there's so many bacteria, so the body wants to get rid of all this bacteria, and it does something very unique to the bladder. They shed in mass. All the epithelial cells just shed, and they're in the urine. And typically when you see all those cells in the urine, that's when you know you have a urinary tract infection. When you go to the doctor, you look in the urine and you see a lot of these cells, you say, you have a urinary tract infection. And so what the bladder does, it sheds all the superficial epithelium just at one go. And that way it reduces the bacterial load tremendously, okay? But what I want to tell you is the mechanism involved mast cells. So the reason we felt like mast cells had a role here was we did an accidental study where we were looking at the shed epithelial cells from patients who have UTIs. And we stained them with antibodies to a mast cell granule component, heparin. And we find that all these epithelial cells that were shed had green in them, heparin. This is what we see. So it looked like mast cell granules were inside these epithelial cells. So what was going on? So we then tried to see what was happening to mast cells during infection. And so this is the superficial epithelium. This is the mast cell in a steady state. But a few hours after infection, we see the mast cells all moving up, right up to the epithelium. So again, this is an image of the epithelium from the top. The green are the mast cells, which are underneath, but the superficial epithelium are in red or pink here. And so that's the superficial epithelium. Below them, the blue lining is the basement membrane. And the mast cells are sitting underneath the basement membrane in a normal situation. When you have infection, the mast cells move up. They cross the basement membrane, which is green. They go all the way up, right up to the epithelium. And this is what we're seeing. So mast cells move up. And when we look, we see that 6 hours after infection, you begin to see some of the green leaving. This is the lumen of the bladder. It's collapsed. But the green is the superficial epithelium. And so you begin to see some breakdown at 6 hours. By 12, a lot of the superficial epithelium is gone. All right, so you can see that there's tremendous shedding occurring as a result of the infection. But interestingly, this is caused by mast cells. So if you look at wild-type mice, they have a nice green epithelium. This is the lumen. When you have infection, you see a lot of the epithelium is gone in a wild-type mouse. But in a mast cell deficient mouse, we see a lot of the epithelium still intact even after 12 hours, whereas here they're almost all gone. But in a mast cell deficient mice that we had repleted with mast cells, we see a lot of the epithelium is shed, right? So the presence of mast cells was responsible for that shedding. And how was that happening? So what we did was we collected the isolated granules of the mast cells, and we exposed them to epithelial cells in vitro, and we're looking for cell death, cytotoxicity, because we think the shedding is because of cell death. And when we look, when we put just bacteria alone on the epithelial cells, we see a little amount of cell death. But when we put granules alone, we see a lot of cell death. And then when you mix bacteria and granules together, we get super amount of cell death. So there's a tremendous amount of cell death caused by mast cell granules, and even more when there's also bacteria present. And when we looked at the mast cells, when we looked at the epithelial cells, we see epithelial cells had taken up the mast cell granules along with the bacteria. So the epithelial cells were dying because they were taking up the granules. And then there was also bacteria present in that epithelial cell, and there was even much more enhanced cell death. And we can see here the granule breaking through the membrane that it was initially encased in and degrading. And this is again another picture of mast cell granules taken up. And you can see the red, which is the granule contents, they're leaking out into the cytosol of the cell because the membrane has been degraded. And this then eventually died. However, if we put a protease inhibitor in here, we don't see cell death. Okay, so clearly the mast cell granules are important in death, and it's the protease in there. And actually, we looked at mast cell kinase as a critical protease that's responsible for cell death. So we got, so we put, this is again a repetition of the previous experiment where we put the mast cell granules on the epithelium. We see very nice cell death, but if we put a protease inhibitor, we see inhibition. And so we cloned the mast cell kinase and put TAT, a peptide that allows the kinase to enter the epithelial cell. We see very nice dose-dependent killing. So kinase, once it enters the cytosol of the cell, it can cause cell death, and it's through a clipping of pro-caspase-1. And so you see the mature caspase. And again, showing caspase-1 activity, the more the more mast cell protease there is more caspase-1 activity, indicating that kinase was in the cytosol, was clipping pro-caspase-1, inducing cell death through IL-1 production and cell death through pyroptosis. So what we're saying is mast cells in the bladder was playing a little different role than what we have described before, where following infection, they go all the way up and then release the granules to cause the loss of the epithelium so that the bacteria clear. So this is a very different mechanism of clearance of the bacteria. But interestingly, we saw something very, very odd. So a few hours after the epithelial shedding and so on has happened, and beginning at 6 hours and continuing till about 24 hours, we saw mast cells producing IL-10 in the bladder. IL-10 was produced in large amounts by exclusively mast cells, which was very strange because earlier on it was producing releasing granules to cause cell death and inflammation there. But what's going on here? Suddenly it's producing IL-10. And when we stain, and interestingly, when we look in the kidneys, this is the bladder. Here in the kidneys, we don't see any IL-10 production. It's only exclusively in the bladder. So we're trying to figure out why. And we then found out, I'm not going into the details, but we found out the real reason was what was happening is mast cells were coming all the way up, causing the shedding of the epithelium, as we said. But then it needed to produce IL-10 to shut down inflammation so that the re-epithelialization would occur. Otherwise, the urine here will make direct contact with the underlying epithelium, which is not tight, it doesn't know tight junctions. So it will cause a severe amount of toxicity. And actually the pain that women who have urinary tract infections experience when they have bladder infection is that because of the urine making contact with the underlying epithelium. So the superficial epithelium is gone. So the underlying epithelium is leaky. And so there's a lot of pain. So what the mast cell is doing is producing IL-10 to shut down inflammation so then that re-epithelialization will occur, right? So we showed that that's the case. But there's a downside for that. The downside is because of this IL-10 production, excessive production of IL-10 by mast cells, the capacity of the bladder to evoke an adaptive immune response is dramatically reduced because the dendritic cells that were trafficked into the lymph node were not active. They didn't have any of the activation markers that they needed as they trafficked to the iliac lymph nodes, right? So here's a situation where you infect multiple times the bladder, just causing cystitis, we do not get much antibody. But if you give a mouse with sufficient infection so that you get kidney infection, you do get very high antibody levels. So in the bladder, you don't get antibody levels. That is because of the mast cell producing IL-10 to shut down inflammation. And this is actually what happens in humans. In women who are the main sufferers of urinary tract infections, if you have just bladder infection, you don't get antibodies produced. And so these women get, again, infection and infection and infection because there's no memory response. And that's because of what the mast cell is doing. It is shutting down inflammation, and so you don't get that response. So I will go quickly to the last part of my talk, which will be very brief, just to remind you what happens with mast cells when they are sitting on the vasculature, that they have a role in immune surveillance of the vasculature. So this is a picture of a mast cell. This is a blood vessel. The mast cell just got activated because it made contact with something some agent in the blood, and you can see all the granules sitting on the outside. And this is a picture of the vasculature and with mast cells just lining it. So in our bodies, mast cells are doing this. They're just sitting in the vasculature. And what I want you to point out is this. So what I've said earlier is when you have an infection, say in the skin, and the pathogen gets in, the mast cell sees the pathogen, it degranulates, it brings out neutrophils and other immune cells to kill off the pathogen. So you have localized mast cell activation. That is good. However, if the infection is pretty severe and the bacteria are able to enter the blood vessel, then a huge difference in mast cell behavior occurs. So what happens is the vasculature is now full of bacteria, which are now flowing through. So because mast cells are lining the vasculature, as the bacteria are going through, or the virus, you get activation of mast cells throughout the body at the same time. So you get this activation, and so you get vascular leakage throughout the vasculature, dilation of the blood vessels, drop in blood pressure, and sometimes you get shock. So when you have viremia and so on, you can potentially have it. So for example, with COVID and so on, there's a lot of virus in the blood initially. So that's enough to activate the vasculature, and that's enough to get the mast cells going. And I just want to point out what happens when mast cells in the blood, when the pathogen is in the blood and mast cells in the surrounding tissue all throughout the body are activated, they can cause tremendous vascular leakage. And in a viral infection such as dengue, which happens mostly in Asia, you can have severe vascular leakage and even death. And so this is what you see. This is where we injected dye, Evans blue, into the blood and looking for vascular leakage. So this is the kind of leakage you get with dengue virus. And this is just images showing the degree of leakage. And these are various organs. And but if you have antibodies against the virus, which are non-neutralizing, you actually get even a worse leakage. This is what happens in many cases in Southeast Asia, where you have the antibodies there causing more severe form of dengue. Interestingly, you don't get any leakage when in a mast cell deficient mouse, whether you have antibody or not, right? So clearly the virus is activating mast cells, but with the presence of antibodies, even a more potent response. And so you get severe leakage here. But without mast cells in the mice, there's no leakage. The animal is protected from leakage. And actually in the wild type mouse, if we can put chromalin in there, you can reduce leakage. So that's a mast cell inhibitor. So I'll just end by saying very briefly that how do mast cells see the pathogen in the blood? So recently we showed there is a dendritic cell which lines the vasculature, which is sitting next to the mast cell, which is sampling the vasculature and passing on the antigen. So we did this with allergy, IgE-mediated anaphylaxis, that there's a dendritic cell that samples the blood antigens and delivers it to mast cells in the extravascular space, and then you get mast cell activation. So I will end by saying there are many phases of the mast cell. Mast cells promote recruitment of immune cells to both the sites of infection and to the lymph node. Targeting of lymph nodes with artificial mast cell granules are highly efficacious in boosting the immune response. And mast cell in the bladder reduce bacterial load, but also then help regeneration of the epithelium. So there's a pro-inflammatory response followed by an anti-inflammatory response. And then the blood-borne pathogens have the capacity to activate perivascular mast cells throughout the body, leading to viremic and septic shock. I will end with acknowledging to all of my trainees over the years. I've run out of time, so thank you.
Okay, so this is really wonderful, and thanks so much, Soman. And I think the audience, including myself, learned many more new faces about mast cell. So now we have time for a few questions. If you have any questions for Soman, please indicate in the chat box. You can either type in your question or directly ask the question by yourself. So we have a first question, I think came from Chuan.
Hey, Soman.
Yes.
This is really great. I'm actually particularly also interested in mast cell because I'm actually studying a lot of excitosis. Mast cell is the perfect model for studying the excitosis. So I think first question I have is regarding the first part of the TNF packaging. So yes. I want to ask about the first wave of TNF degranulation happening within 24 hours, if that's correct. Yeah. Okay. So sorry. Go ahead. No, go ahead.
No, I just want to say so there is a pre-stored granules. I mean TNF in the granules. So there is that release, but then there is de novo synthesis of TNF, which continues for 12 hours at least.
Right. So because you mentioned that it's actually travel all the way to the lymph nodes. So my first question is how, if you can actually have any comment that how the granule get degraded so the TNF can be released in the lymph nodes?
Yeah, so we don't have the exact answer for that, but I think what happens is the granules are trafficked passively through the lymphatic vessel. They get to the lymph node. Then the lymph node, they have heparinases that break down, they attack the heparin. And then that's what we think is the critical release. So it gets to the right place, which is the lymph node. But in the local environment of the lymph node, the heparinases then chop up the heparin to release the granules. And so after a few hours after infection, we see that the granules slowly, they are in the lymph node, but then they slowly disappear.
So basically, you actually can manufacture a machinery that control the degradation of the membrane of the granules, so you can control the release of the substance, if I understand this correctly.
No, okay, so this is very key. There is no membrane around the granule.
Yeah, sorry. Yeah, no, that's true.
Yeah, so there's a membrane inside the mast cell that keeps the granule, but once they come out, it's an electrostatic interaction.
Sure. No, I think what I mean is if there's any way you can manufacture a machinery that you can manipulate the release of the granule, so you can actually control the location and time of the release of the substance.
Yes, that's a good point.
The other thing which I actually am curious is whether this release of the granule are particular to the TNF or the other substance. We know that mast cell also a huge storage of the histamine. That's also a part of the whole machinery, or it's actually particulars for TNF?
Okay, this is a very good question. So what we are seeing is so there are a lot of things contained in the granule, but when the granules are released, some of the most soluble ones like histamine come out first. They are within a minute or two, almost all the histamine is gone. It's like candy there, whatever dissolves first or that goes first, but the TNF are held much tighter. So there is a gradation in what is coming out. So this is a slow process, and it's like candy. The granules we see sometimes getting smaller and smaller. And so we think that's what is happening. So there are some with low affinity for the granules, they come out first. Then those with tighter affinity stay longer. So I think that is what we think is.
So you can either even synthetically manufacture a granule which actually can have different gradient to release it. But is this actually depends on the molecular weight, or is something else?
So everything is charged. So we think it has something to do with the charge that they have, the intrinsic charge, because these two are held together with charge. So I don't know about the molecular weight. I mean, TNF molecular weight is pretty small, but it's still held. So it's possible. I don't have the answer.
I see. And then when you actually mention, when you actually synthesize this granule in vitro, have you ever tried, in addition to synthesize the whole particle, actually have you even considered to put multiple substances together in this complex, so you actually can have multiple functions?
Yes, potentially. So for example, we put the particles, when we put the antigen in one set of particles and the cytokine in the other, and then mix them together, so that because we want the particles to be taken up by dendritic cells, and then the cytokines and so on, we want it to be broken down and taken up by other cells in the lymph node. So by mixing and put them in the particles and then put them all together, you can actually direct them to different, based on their tasks.
Right.
So that idea is possible, but we haven't done it.
Right. Okay. My next question is regarding your second part of the bladder. I found pretty fascinating. So my first question is, why the bacteria actually can induce mast cell to migrate into the epithelial cells across the membrane, matrix membrane.
Yeah, so we've shown it is actually IL-1 beta. So the epithelial cells, they produce IL-1 beta, but they don't die. When they have the UTIs, so they produce that. And that IL-1 beta is what causes the mast cells to go all the way up. So that's actually the cytokine responsible for taking them up across.
I see. But regarding to this, because when you're describing that when the granule was uptaken by the epithelial cells, it's actually can induce peritosis
Yes.
for shedding the bacteria. So whether this actually also occur in the other epithelial barrier surface, like in the GI?
Yeah. So I don't know. It's possible, but in the bladder, unlike in the GI, you lose one epithelial cell, another one there, another one. But in the bladder, for some strange reason, they all go out at the same time. It's just a massive exfoliation. And I don't know, we've never seen any part of the body except in the bladder. So they may have it isolated one, but this one.
Yeah, I was just really curious if there is infections in the intestine, whether the similar.
It's possible. Yeah. Because
Yeah,
I think now we're learning mast cells can move into right up to the epithelium.
And this process, I assume, also occur. I thought you say within 12 hours. The particle uptaken.
Yeah. The shedding.
Yeah, the shedding. It's happening within.
Yes.
Very sure. Huh.
Yes.
Okay. Thank you very much. Thank you. That's interesting.
So
okay, good. I think there's also she also raised a hand. Maybe we can.
yes.
Hey, Soman, this is really cool talk. I have a couple questions. One similar to actually to Tran's question regarding the packaging of those particles and how they're being released.
Yeah.
Remember you mentioned in the synthetic particles you have, you seem to pack them in acidic pH, maybe to increase their binding or sequester of the cargo. And do you know if in the actual particles, the granules produced from mast cells, whether the binding is pH dependent?
It's a good question. So the way we make these particles is we incubate these at low pH, the cargo and the negative and positively charged particles, and they're all there. And then we raise the pH, and then they all come together in nice spherical shape. But we've not tested the natural ones to play with the pH. But that's an interesting question. But my guess is it'll behave, when you adjust the pH, it may behave very much like the natural, like the artificial one, but we've never really tested that. But from the literature, they always say that it is, helped by electrostatic interactions. And so some of the classic biochemists have done a lot of work. I can't remember.
Curious if in the acidic environment, maybe where this particle is trafficking to the lymph node and it's being actually released when exposed to a more acidic environment. That's probably also when some of the enzymes become more active.
Yes.
At those locations.
Right, that's a good point. Yes, I didn't think of that.
And in terms of the hyperinase, is really fascinating, the mechanisms you described. Do you know where in the cellular space are the hyperinase expressed? Are they membrane anchored or are they secreted?
Oh, I have no idea. Yeah, no, we've not gone that far.
Oh, cool. And linking back to the question that you showed really nicely, the TNF in the particles seem a stronger potency than the soluble form TNF.
Yeah.
Do you think that's due to the avidity of the particles, or is due to potentially signaling through R1 versus R2 of the TNF receptors?
Oh. I don't know if it has anything to do with the TNF receptors. I was thinking more in terms of the stability of these particles. I didn't think, but it's possible. They are more potent because when there's TNF and there's some sort of, it might cross-link the receptors and there are more potent signals. I mean, like people have described a long time ago that TNF expressed on the surface is a much more potent stimulator of the receptor than the soluble one. So that has been shown, but I don't know in this particular case, but it's an interesting thought. I don't know the answer.
Well, and my sort of last question, the location of mast cells is really fascinating, right? It's right next to blood vessels. And it's your thought that it's surveying more on the tissue side or potentially also surveying on the blood lumen side as well? Because in many cases, some of the antigens activating anaphylaxis may not be able to penetrating the blood barriers, but they may be able to activating mast cells degranulation.
Yeah.
Do you know if mast cells are able to sample even the antigens on the lumen side of the blood vessels?
Right. So like I was saying, maybe I was rushing it, but there are dendritic cells that sit and they stick their pseudopod-like thing and grab from the blood the antigen, and then they take it in and then they pass it on vesicles. So the granule, the antigen is on vesicles which bud off, and then the mast cells are sitting just next to it. So the vesicles then cross-link the mast cell, and then you get anaphylaxis. So there is a professional antigen-grabbing immune cell sampling the blood at all times. So even if there's no antigen, we can see the pseudopods of the dendritic cell sampling the blood, just like that. But the moment it catches something of interest, it binds. So we found the scavenging receptor is very important. The mannose receptor is important in picking up antigens on the surface of dendritic cells, and then it gives out vesicles to the mast cells.
Cool, thanks.
Yeah.
Okay, so we have a few questions in the chat box. I think first one came from Zhao Jie. I probably just read the question here. What induced IL-10 production in mast cells to shut down the inflammatory response?
Yeah, that's a very good question. We've been trying to mimic it in vitro, like what is produced, but we think it's very unique to the bladder. That's something, and we don't know what the signal is, because when you infect the kidney, we don't see that at all. We have bacteria in the kidney, we have infection, but no IL-10. This IL-10 is produced specifically by the bladder mast cells. So what stimulates it, we are yet to find. We've tried through in vitro work, so many agents, but we don't see that.
So I wonder if there's any, let's say, single cell sequencing has been performed in the mast cell, in the bladder versus in the kidney, or the other possibility is the interaction between mast cells with the epithelial layer in the bladder. Probably there's some like
right. It's possible. We are currently trying to do single cell sequencing at different time points during infection. So maybe we will learn something, but we just started that.
I see. So maybe a related question here is the origin of these tissue mast cells. So are they like always there, or they can actually came from some of the bone marrow or spleen?
Yeah, so the mast cells in each site of the body, they say, is different and is based on the surrounding cells. So they come in as immature cells from the circulation. So they get in there and then they mature in the site. So when they mature, the surrounding cells dictate how many granules or what is the composition of the granule and so on. And what we have found is after, say, an infection has occurred and now the mast cells are regranulating. So now it is regranulating in a place that had an infection, which is somewhat inflamed. So the contents of the granule there is quite different from the naive ones. So the surrounding is a very, very key thing in how the mast cell behaves subsequently.
That's super cool. It's a context dependent, like granule component synthesis.
Yes.
Okay, good. So the other question from Chen Ke, is there any difference in gram-negative and the positive bacteria infection?
Yes.
I guess.
So the way I have talked about is like all bacteria are the same. No, that was for simplicity, but there are very, very different behavior. So gram-negative, there's some gram-negative that are very potent activators. Some are actually inhibitors. So we showed like Salmonella, which is shown here, is a potent pathogen that can shut down mast cells. It will bind and they inject a phosphatase, which will prevent mast cells from degranulating, but it does not block the bacteria from entering the mast cell. So it can paralyze the mast cell by producing this phosphatase. And the same as Yersinia pestis, this is the pathogen that caused the plague. So both of them can shut down mast cells. So even among gram-negative bacteria, there's a huge difference. Now, gram-positive, there's some like Staph aureus that activate mast cells very well. Then there are others that less. So it's very different. So it's not a general thing. Every pathogen, and then in some cases, if there's antibody there, then the antibody will coat the bacteria. And so their interaction with the mast cell is through Fc receptors. So it's very, very different depending on the pathogen, the environment, the host immune system.
Okay, cool. Okay, that's great. I think this is a very nice discussion. And if there are no more questions, let's thank Soman again for this wonderful talk. And personally, I enjoy a lot. I think that's also the case for this audience. Thank you, Soman.
All right. Okay. Bye.
Thank you so
Bye.
much.
Bye. Thank you.